So I'd like to thank you for this opportunity and for having me. I'm really pleased to talk to you all. Today I'm going to be talking about diversity. So, first of all, I'm enrolled in a graduate program at the Museum of Zoology of São Paulo under the supervision of Cristiano Scherner and Pablo de la Pe. Cristiano Schrettner. Sorry, Cristiano. And then let's start the talk. We'll be talking about diversity of city bugs, but what kind of aspects of diversity we are talking about? So I'm giving you some aspects of biodiversity here. So on the left, we have some shortfalls of these aspects of biology, biodiversity. For example, Linnean shortfall means that we have a gap of the knowledge about the species, or like the identity of the species. Another example would be Wallachian shortfall, and that would be corresponding to the geographic distribution. And we can go to other aspects like populations, evolutions, and so on. But let's keep in mind that the first two of these many aspects are really important to us because they critically affects all of the others. Okay, so if you don't have the idea what is out there and how they are distributed, it's hard to ask other further questions. So where are we at? So it depends. Let's take as example lions in Africa. They have pretty much a good map about it. They even have a historic range of these species. Okay, you can say that they just one species or maybe two subspecies. It's okay, it's easy to track them. But okay, let's talk about trees in Europe. These are an example of predominant European trees. So we have another map, they can be explored. And okay, but then we can talk about, say that the trees are fixed, they don't fly, they don't swim. Okay, so then let's talk about fish. The freshwater fish. This map shows us the global distribution of freshwater. So the more red we have, the more information we have about the species, which means that they're reaching virtually all the diversity in that locations. Okay, and how about insects or Heteroptera, Hemiptera? I'm bringing this this example of a study that has been just accepted. That here we did a first survey of new species published in Brazil in 2020. And we can see here, like as an example, Hemiptera has 89 species published in 2020. Okay, so if we keep that pace throughout the other years, we have here in this right table the estimated time to reach the estimated diversity that we have in real life, let's say. And then we'll take probably 224 years to reach that number. Okay, so it's a lot of work, of course. So here it's a good example of a Heteroptera in Brazil, led by Dr. Felipe Moreira. Has been published with his team out of the checklist and distribution information about the semi-aquatic and aquatic true bugs. He also shares a distributional database that contains plenty of localities, that more than 1,000 localities here in Brazil. And we plot that in a map, we can see a pretty much full map, right? So that's a good example for us. So here in this map here, I have like some colors, and I'm going to explain now. These colors are the 6 ecoregions that we have here in Brazil, which are Amazonian rainforest, Pantanal, basically wetlands. We have the Cerrado, which are our savanna. We have the Caatinga, that it is the driest forest here in Brazil and the poorest explored ecoregion for like diversity studies. We have also the Atlantic forest, Pampa, that's basically a grassland in the south of Brazil. This diversity of ecoregions gives us at least 10% of the global diversity. And some authors argue that's even more, like 15 to 20%, but still it's a lot. Okay, Rhyparochromidae, the group that I'm working on, it's in the superfamily Lygioidea that has 16 families worldwide and 11 neotropical families that are listed here below. And here you can see that's pretty much a diverse group. And here are the Rhyparochromidae that I'm working on right now. So Rhyparochromidae has more than 2,000 species worldwide and 84 in Brazil, which is less than 10% of the global biodiversity described. We have also 14 tribes and 8 tribes in Brazil. The family is recognized by presence of head trichoboltrias and abdominal sutures. In the 4th and 5th here, right here, they do not reach the dorsal margin. For the first glance, we can ask like, oh, are they predators? But they are, but from of seeds. That's called that's why they are called seed bugs, right? So this forefemur is really robust to grab and grasp it and handle well the seeds while they're eating. They also consider pests sometimes, and they have economic importance. This example, this Hemiptera bilobata here, it feeds on strawberry. It's a classic example. It feeds on the seeds of the strawberry and then cause damage on the fruit itself. Until to this day, catalog by James Slater from published in 1964. It's really important for us to know the distribution and all the information that we have on the superfamily. And in 1994, Slater and O'Donnell published a complementary work for that. So online, you have the other sources like this Lygioidea species file ran by Pablo de la Pé, Thomas Henry, and David Eads. And for Brazil, we have the taxonomic catalog of Brazilian fauna. We have it is updated by experts of all organisms here in Brazil. So for Rhyparochromidae, there's no survey for Brazil. There are only a few specialists dedicated to the taxonomy of this group, and no previous Brazilian taxonomist working on this group. And then there is only one species described by a Brazilian in 1960, 1956, sorry. And here we have this book published in 2012. That's an important textbook for undergrad and graduate students. And now a new edition is coming soon with updating information, families, keys, and making sure that all the information about the Rhyparochromidae and like Ligoidea it's there in there. So remember that that ecoregion that I was talking about, like the Caatinga, which is dry forest. We are in a project. We are developing a project there. They're trying to collect for the first time Hemiptera in that region. And I was lucky enough to participate in their collections as well. We use different methods, myeleza, fly traps, sweeping, even water bugs too. In addition to that, I have visited some collections in Brazil as well as abroad, trying to access more data. But when I visit those museums in Brazil, it's the Rhyparochromidae or even Ligoidea is rarely identified due to lack of experts, literature, and we don't have other specimens for comparison. That's a challenge for me, I think. So what I'm trying to do here is to describe, quantify, and map the knowledge of biodiversity you have here. So in that, I have a focus group that I work on a tribe Latin Rhyparochromidae. And let's have an example that I've been doing here. This species, Petis spinops, was described by Stoll in 1874 in Guyana. Okay, and then 2005, Boronowski and Slater published a work on the West Indies, and they found this species in certain Caribbean islands there. So it makes sense. They're pretty much close. But then 2015, Pablo de la Paix published the revised Lithuanian for Argentina, and they also found that species there. So we have a wide gap there. So in my visits and in our field trips, we have collected a bunch of them. So we filled a gap here. So that's the goal, like to try to understand the distribution and then ask more questions. Here, it's the species that it's some examples of the species that have been collecting and seeing in the museums. So you can see that all of them are new to country or new state record. Some like here, so even for genus like Libostomata, it's new to the country as well. And when you have state records, 2, 3, even 7 states new records. But we still have much more to do, of course. We have potential new species, and then we need to. We are still in the Linnean and Wallacean shortfalls, and now we are getting back to the lab in collections. We can work on the species in person, which is great. And always when I see our vials from the collections, it's always full like this. It's a quite complex group for me, and it's pretty diverse. That's pretty much interesting things there. And all samples are stored in ethanol, and that's allow us to access other data, such molecular data, to make phylogeny, also, as well as morphological phylogeny. Yeah, and then I hope to see you in Barcelona and to present you more data. And thank you very much for the attention.
Okay.